Okay, so good afternoon. Today we shall be discussing the first part of the gram-positive bacilli or the aerobic gram-positive bacilli. Okay. So for the aerobic gram-positive bacilli are divided into two major groups, whether they are spore former and the other group is the non-spore former. Under the spore former, meaning to say they have spores, okay, the most common of them all would be the Bacillus, and there are several pathogenic species under the Bacillus, such as the Bacillus anthracis. Okay, the difference with that of the other group is that Bacillus anthracis are non-hemolytic, and then the other species of Bacillus are considered to be hemolytic. Okay, but the most pathogenic beta hemolytics and motile species of Bacillus is the Bacillus cereus. Okay, now under the non-spore former, we have the aerobic actinomyces, Arcanobacterium, Corynebacterium, Erysipelotrix, Gardnerella vaginalis, which by the way could cause vaginosis, Lactobacillus, and Listeria. So how do we divide them? They are being divided whether they are branching and the other one is non-branching. Branching in such way that some of them could be mistaken as fungi, such as the actinomycetes and nocardia. Nocardia, noteworthy to mention about nocardia, it is the only species aside from Mycobacterium that would have mycolic acid. So nocardia is slightly acid-fast organisms. Okay, most of the bulk for discussion for today are the non-spore forming, non-branching gram-positive bacilli. So under that category are the Arcanobacterium, Corynebacterium, Erysipelotrix, Gardnerella, Lactobacillus, Listeria. So how do we divide them? They are classified whether they are catalase positive or catalase negative. You know what we mean by catalase, right? We utilize hydrogen peroxide, and a positive reaction is characterized by rapid effervescence of gas or bubbling. Now, catalase positive are the Corynebacterium and Listeria. So this group is further divided by means of motility at 2 degrees centigrade and bile-alkaline hydrolysis. Listeria is motile at that temperature, whereas Corynebacterium is not motile. Okay, those that are catalase negative, okay, they are further divided into the ability to produce hydrogen sulfide. How do you know if there's hydrogen sulfide? You have your TSI. If there's a blocking in TSI, so that's considered as positive for hydrogen sulfide production. So the only one that is hydrogen sulfide positive is Erysipelotrix. So the rest are actually considered as H2S negative. Okay, so that's how we classify the organisms. Okay, for the aerobic gram-positive bacilli. If you want to have a clearer version of this one, you go to your book main one, figure 16-1. Okay, so let's discuss first the Bacillus species. What's quite unique about Bacillus species that they are found in nature. In fact, if you're one of the antibiotics that we have is bacitracin, right? Do you remember bacitracin came from what species of Bacillus? Bacillus what? Yeah, that's polymyxin. Pag bacitra, yeah, bacitracin, that's another species of Bacillus, right? So they are found in soil. They are found in nature. The most common lab contaminant is Bacillus subtilis. If you're not careful with your work, you'll be able to see large colonies of bacteria that whitefish. So that's Bacillus subtilis. So they are widespread in nature. Most of them are saprophytic, Okay? Some even are isolated as contaminants. Okay? So they are gram-positive, empty, with empty spaces. What do you mean by empty spaces? I'm not talking about your heart, okay? I'm talking about the endospore. So it is as if there's a big hollow space, okay? There's a big hollow space. So this actually represent the spores, okay? The spores, okay? So the endospores. The endospores are highly resistant to heat, drying, and chemical. Okay, and they are also catalase positive. So this is how they look like in sheep's blood agar. Bacillus cereus is considered to be as beta hemolytic. All of them are capable of hemolysis. They are motile except for Bacillus anthracis. Okay, so why they are motile? Because of the fact that they are being provided peritrichous flagella. So when we speak of peritrichous flagella, as a review, the flagella surrounds the entire organisms. Okay? The cell wall is also noteworthy to mention. The cell wall is made up of mesodiaminophenolic acid. So that's a unique constituent in the cell wall of Bacillus species. Okay. We classify them according to the endospore formation. So, let's say for example, group 1 has ellipsoidal spore. Okay? Group 1 has ellipsoidal spore, but the spores of group 1, okay, is further divided into two, whether you have a large cell or a small cell. So ellipsoidal spore is somewhat located here. Okay? So, such as Bacillus anthracis, the one that can cause anthrax, cereus, megatherium, Bacillus thuringiensis. Perhaps this is one of the most popular species of Bacillus. Okay, particularly being used as a vector in GMO or genetically modified organisms. Okay, so the plasmids of a certain genes, okay, the genes that contains the desired trait that you want a certain plant or organism to have would be incorporated in Bacillus thuringiensis. So it serves as a vector. So there you can have a corn that is very yellow, very large. So there you have now what you call the BT corn. The controversy regarding the GMO is that it's being controlled by certain corporations. Monsanto, if you're familiar with that. So it's being monopolized by certain. So farmers are actually at the mercy of Monsanto because they are the only ones that can provide seedlings. Because the seedlings contain the genes of a desired trait. Let's say, for example, you want rice that can withstand flood. You want rice that is anti-pest or pest resistant. Or you want corn that is big, very sweet, because they incorporate all of the genes, a certain trait in a certain plasmid, and then they place the plasmid. They utilize the plasmid of Bacillus thuringiensis. That's why they call it BT corn. And another controversy is that if the consumer or us consume these GMOs, we do not know what will happen to us because there hasn't been a thorough study. Well, there might be a good probability that it could be an X-Men in the future. Then you choose the power of your choice. Ako na si Professor X, ha? Wala nang kukuha na. Ako na si Professor X. So yun yung mga possibility ng GMOs. That's why it's still very controversial. So they utilize Bacillus thuringiensis. Subgroup 2 means that they have smaller. They still have ellipsoidal spores, but the cells are smaller, less than 0.9 microns. Okay? And then group 2, oval-shaped spore. Bacillus theaterothermophilus. Are you familiar with this? This one is being used as an indicator in the autoclave. If you want to check whether the autoclave has been effective or not, then the indicator organism, biological indicator, we call it biological indicator. So that's Bacillus thermophilus. Yeah, Bacillus pollinixa is the source of your pollination. So how do you characterize the spore? The spore is oval shape and it's swollen. Okay, it's swollen. Okay. The other one that is swollen circular. Okay, swollen circular. Circular talaga and swollen. Okay. So that's group 3, Pastöris, Sphericus, and Pantotechnicus. Okay? So Bacillus species generally do not grow in CNA agar. Okay, so to identify, we know for a fact that they are catalase positive, they are buds per shower positive, they have the ability to grow in a jar that is filled up with oxygen and at this temperature, 50-65 degrees, because of the fact that they have spores. So they can withstand much higher temperature. Okay? They can also survive in a medium that contains about 7.7% salt. They have nitrate reductase, an enzyme that reduces nitrate to nitrite. They can hydrolyze starch and they can decompose casein. Okay, so these are the characteristics of Bacillus species. So let's start discussing one of the most significant species of Bacillus anthracis. We actually asked 100 terrorists and asked them what's their favorite organism. Survey says, parang ano lang kami cute. Number 1 in their list is Bacillus anthracis. Okay, you know why? Because Bacillus anthracis can actually be used in bioterrorism. Maybe I'll explain it to you later. So it's a large, encapsulated, non-motile, and it appears like a disjointed bamboo. So this is how they look like, and they are called the medusa head colonies. You know what medusa is, right? In mythology, this is a girl with snake head, right? So in liquid medium, they appear as the inverted fir tree. Okay, it can cause the disease known as anthrax, black beard, or Siberian fever. And these are also encapsulated. In fact, the capsule is made up of glutamic acid. Okay, they also have the cell wall is also made up of polysaccharide somatic antigen and virulence factors known as the anthrax toxin, such as the made up of protective antigen, the lethal factor, and then the edema factor. So these are the virulence factors of Bacillus anthracis. Okay, so this is the disjointed bamboo appearance of Bacillus. So you'd notice that there are empty spaces. Okay, empty spaces. So those empty spaces are actually the unstained part, because if you want to stain it, you need to have a special stain for spores. Okay, so these are actually the empty spores, and this is the medusa head colony. So if this is the colony, it is as if that there are structures in the colony that makes them like the medusa head. And if you notice, unlike your E. coli, S. aureus, the colonies are actually bigger. And it's mucoid because it's made up of capsules. Okay, so Bacillus anthracis is actually a disease in the farm, because in the farm, particularly the soil, in the fur, okay, spores are found there. So humans get accidentally infected because of the contaminated wounds or ingestion or inhalation of spores. So take note that there are 3 possible ways by which you can get the spores of Bacillus anthracis. So it's the large spore-forming gram-positive bacilli. This is one of the reasons why it's quite very popular among the bioterrorists, because the spores are viable even if it's kept for more than 50 years. So imagine they just place it in an envelope, and when you open the envelope, there's a chance for it to be muted. So another reason why it's favorite of terrorists. There are three ways by which you can get it. And the easiest way for humans to get it would be by means of inhalation. So, they place it in the powder, and then you open an envelope with powder, the powder puffs in the air. So, once the powder has actually puffed in the air, there's a chance for you to inhale the spores. And the mortality rate is very high if you get it by inhalation. So, that's one of the characteristics. Okay? Vaccination, is this available? Yeah, particularly for those individuals in high-risk exposure. Who are the high-risk individuals? Well, most of the time, those who work in the farm. So, these are the individuals that are high risk. Bioterrorism, not as as of now, there's no danger, or we don't know. They usually attack by surprise. Diba? They usually surprise us during their attack, so we cannot tell. But those that are high risk are those who work in the farms. Particularly, we call them the wool sorters. Okay, wool sorters. They are the ones who sort the wool of the sheep, because this can also be used in textile industry. Okay, so I did mention to you that there are three ways by which you can get the spores. Pulmonary, you get it by inhalation. Gastrointestinal, if you ingest the infected meat or contaminated water or maybe unpasteurized milk with the spores. Even if you cook properly the meat, there's still a chance that you'd still get it. Because remember, they are made up of spores, and spores are not destroyed by heating. Okay, and then the cutaneous part is that you have an open wound. Okay, and then the open wound has been contaminated by the spores. Okay, so because of that, we have actually three types of anthrax. So we have the cutaneous anthracis. So ito yung sinasabi ko kanina, you get it by, you have an open wound, and then your open wound that contaminated by the soil that contains the spores. Okay? Or you touch contaminated meat, and somehow or one way or another has created an eschar or ulceration in your skin. Pulmonary anthrax, known as the wool sorter's disease, those who are sorting wools might actually get contaminated because the spores are either in the wool of the sheep. Okay, and then intestinal anthrax, the mode of transmission would be by means of ingestion. Take note, number 2 and 3, they have high mortality rate, 90%. 9 out of 10 people would actually die. Unlike cutaneous anthracis, 2 out of 10. Okay. Fortunately, the incidence of infection is very low, fewer than 5 cases per year. Wool sorter disease are also known as the rag pickers, those who handle fibers, hides, or other animal products. They are the people who are at greater risk of getting bacillus. Okay, but most of the infections are cutaneous anthrax, either through cats. So if you have cats, this will cause localized infection, and then there will be a malignant pustule. Okay, so you call it black eschar or the eschar formation. It's painless and it doesn't produce any pus, but the bad thing about it is that once you have the lesion, it will be a permanent thing in your skin already. Okay, so yeah, that thing is permanent already. Well, maybe, I don't know if it's permanent, maybe you can do something about it. Get a tattoo and design the sands. Diba? So, at least, diba? At least, it becomes artistic. It becomes permanent, diba? Or maybe, here, you can make an Aztec design. Aztec design, no? Diba? Aztec yung design ng tattoo mo, diba? Astig. So, something like that. So, at least, if you have a permanent scar, you cannot tell, oh, it's a different tattoo. 3D embossed. That's right. Yeah? Sino yung artist mo? Well, I'm so sorry. You cannot actually get it. Masyadong pinaggastos sa itong tattoo na to. Masabi na gano'n. Okay. So, gastrointestinal anthrax, how do you get it? You either acquire ingested You acquire it by ingesting contaminated milk, or it has been inoculated into a lesion on the intestinal mucosa. How would you do that? Of course, by ingestion also. Okay? So, signs and symptoms would include abdominal pain, nausea, then anorexia, keep on vomiting, and sometimes you have a bloody diarrhea. Okay? It's actually more fatal than the cutaneous anthrax. Okay? So, violent anthracis. It's mostly a disease of an animal. Okay? The pulmonary anthrax is also known as the wool sorter's disease. So, you acquire it by inhalation. Take note, the incubation period is very short. Only about 2 to 5 days. And you might be thinking that it's just an ordinary flu or common cold that lasts about 2 to 3 days. Suddenly, you'll have respiratory distress, disorientation, you'll be comatose, and eventually die. So that's how sudden pulmonary anthrax is. Okay? And it would last in less than a day. Okay? You would actually die. Okay? So that's why it's very dangerous. So pulmonary anthrax is actually an accidental. Humans are accidental hosts because it's actually a disease of the animal. It just so happens that we inhale the spores, unless it has been introduced to you by a terrorist. Okay? So if you're a medical technologist and you're suspecting anthrax, you cannot just open a specimen, okay, in a bench lab. Okay? If the specimen is not inside a biosafety cabinet. Otherwise, you'll have what? Pulmonary anthrax. What is this? Open mo pa yung plate, then you smell it. Ayan. So there's a chance that you'll get pulmonary anthrax, and we'll expect death in 2 to 5 days because that's the incubation period. Maybe it's a big chance that we can post as Facebook status APF, inhaling spores of Bacillus anthracis, and make a selfie, then post it online. It will go viral. Though the etiologic agent is bacterial. Okay, so it's non-hemolytic, non-motile. They are capable of producing the symptoms. Okay, the strain of pearls. This is one of the lab tests for Bacillus. What does it mean? Once you expose them with antibiotic penicillin, the cell will enlarge. Let's say, for example, this is the original appearance of the cell, but you expose them to penicillin, it will be swollen. So it is known as strain of pearls. Iba yung pearling, ha? Baka ma-associate niyo sa pearling. Ako, iba to. Iba yung ibig sabihin ng pearling. Hindi maganda yung ibig sabihin ng pearling. Okay? Yung string of pearls, yun yung positive reaction. Kaya yung positive reaction for anthrax. Okay. So, lab diagnosis, restrain it with Bacillus and use the flat medium. So, animal inoculation test, not the classmate inoculation test, no? An animal inoculation test is another test. It's a string of pearls which I have already discussed to you. And the Ascoli test, wherein Bacillus anthracis is positive for soybean lectin but negative in snail lectin. Okay. Under the microscope, I have shown the appearance already to you. Appears like disjointed bamboo. But if you're looking at the plate, which I have already shown to you, they are non-hemolytic in 5% blood agar. I keep on telling you this, so hopefully you'll not forget about that. And they have, you've seen a while ago, right? Grayish, whitish, irregular, medusa head or beaten egg whites appearance. So that's the colonial morphology of Bacillus anthracis. Okay, so here again, this one, the spores are not that obvious. Unlike this, is the hollow spaces or blank space. Okay. Okay, so in Bacillus anthracis, so Yan, Bacillus anthracis. So, kung nakakasalita lang siguro yung Bacillus anthracis, sasabihin niya, I have a blank space here and I write your name. But, again, actually, hindi naman talaga siya blank space. Unstained part lang talaga to ng endospores. Okay, puti na lang, hindi nakapagsalita yung Bacillus anthracis kasi parang corny, no? I have a blank space here and I write your name. Sabihin ko lang. Okay, so, how do we differentiate Bacillus anthracis from Bacillus cereus? Why do we need to differentiate them? Because these two are the most pathogenic organisms or pathogenic species of Bacillus. Which one is capable of hemolysis? Cereus. Which one is motile? Cereus. Which one is susceptible to penicillin? Anthracis. Which one can produce Bacillus? Both of them. So association types. Okay. Which one can ferment salicin? Bacillus cereus, but not all the time. Can grow in the presence of penicillin. Syempre, cereus, because anthracis is susceptible. String of pearl positive, anthracis. Can hydrolyze gelatin, cereus. Can grow in PEA, that's phenyl ethyl alcohol agar. So, cereus. Okay? So, are you serious? Yes. If you have this particular characteristic. So, that's Bacillus cereus. Okay. So, but how do we treat anthrax? Fortunately, there has, unfortunately rather, anthrax has developed resistance to penicillin. Well, it's unfortunate that the thing that has been discovered by Alexander Fleming in 1928 has become a useless thing nowadays because of the indiscriminate use of antibiotics. Organisms, bacteria, had actually become wiser. Believe it or not, they have become wiser. They are actually capable of evolution. Evolution means they are able to adapt to changes. And bacteria can do such a thing. So we discovered penicillin, they produce beta-lactamase. You know, they also have researchers okay, among themselves. Oh, humans are producing penicillin. Oh, we produce enzyme that would counteract penicillin. So that's beta-lactamase. So beta-lactamase destroys the beta-lactam rings of penicillin, making it useless. Therefore, instead of using penicillin, so we have produced another cephalosporin. So this time we use ciprofloxacin. So we have now another level of antibiotics. But I don't think that Bacillus anthracis had gone into research of how they would combat ciprofloxacin. But you know what? It is us humans that are at fault. Okay, why these bacteria develop antibiotic resistance? Because of indiscriminate use, because of lack of knowledge. Okay, so some people will drink antibiotic for no reason at all. Remember the post that I did? A woman would buy erythromycin for just 2 days, and after the symptoms have subsided, she'll stop using erythromycin. But the problem is, she wasn't able to eradicate bacteria. And those who were able to survive would eventually find ways. Okay? Sabi ng bacteria, we find ways. Okay? So, they would find ways on how to combat erythromycin. So, eventually, erythromycin of that stupid lady Mercury, okay, will not anymore be effective. So, the next time she'll have the same signs and symptoms, most likely she'll buy erythromycin. But that erythromycin will not anymore be useful for her. See? And the problem is, here in the Philippines, pharmaceutical companies or pharmacies are dispensing drugs even without prescriptions, even without pharmacy counseling. That's very important. I've been to several countries. Let's say, for example, I need to buy, when I was in Singapore, I need to buy a mucolytic. Mucolytic, they don't have Solviox, but they have a different carbocysteine. So I have to buy it, but they won't give it to me because I'm not a Singaporean. I don't have any prescription. But I told the pharmacist, you don't need prescription for mucolytic, for carbocysteine. Tapos umubo-ubo, is it obvious that I have? But may kasama ako. So what she did, she actually got the permanent ID. They call it they have this S-Pass if you're a Filipino and you work in Singapore. That is S-Pass. So they got the S-Pass of my companion. They log it so that they have a record that her companion actually acquired certain amount of carbocysteine in that pharmacy. So pag nag-addict ako, siya yung kukontakin. Parang gano'n. Ganun kahigpit, di ba? Ganun kahigpit sa ibang bansa. When you go to China, when I was in Beijing, I need to buy drugs. Hindi pala uso sa kanila yung 3 tableta. I need to buy antacid. When I bought an antacid, binigyan ako 1 box. Yes, bawal yung 3 lang po, 3 tableta. No, 1 box yung binigay sa akin. Oo, 1 box, 1 panig, gano'n. Pag bumili ka ng antibiotic sa kanila, ibibigay sa iyo 21 because you have to drink it 3 times a day for 7 days. Or depende sa antibiotics. Bibigay sa iyo yung entire dosage. Hindi pwede yung bibili ka na lang ng antibiotic. But because they're actually using it to prevent antibiotic resistance. Okay. So let's talk about Bacillus cereus. The second most virulent species of Bacillus. And there are, what strikes me here is that there are 2 types of enterotoxin. By the way, what is an enterotoxin? When you say enterotoxin, it is a type of toxin that targets intestinal cell. That's why it's called enterotoxin. If it's a neurotoxin, it targets on your nerve cell. Okay? So there are 2 types of enterotoxin. One is heat labile and the other one is heat stable. What's the difference? When you say heat labile, it is unstable in the presence of heat, which means that you can easily destroy the toxin by applying heat in your food. The other one is heat stable, meaning cannot destroy the toxin even if you heat the food. Okay? So imagine these 2 toxins. Therefore, Bacillus cereus can actually contaminate and cause contamination and can cause illnesses in 2 types of food. Whether you have a cold food or even a hot food. Let's say, for example, there are food that cannot be eaten. Let's say, for example, cold cuts. That's why it's called cold cuts. We're crying out loud. Diba? Kaya nga siya salami is an example of a cold cut. So you don't eat it. Salad. Diba? You don't hear it. You serve it chilled. Diba? So, which means heat labile enterotoxin can actually infect. Because you cannot order a piniritong salad or a roasted salad. Diba? Fruit salad nga po yung boiled. Diba? Wala naman ganon. Or, paano naman yung heat stable? Usually sa mga rice meal to. Sa mga rice. Kapag improperly, kapag na-contaminate, diba, nasa soil sila. Nasa soil. Tapos, since nasa soil sila, alam mo yung mga rice na kailangan mo siyang iprito or sinain mo, yung rice, diba? Tapos yung kanin na fried rice, buhay pa rin sila ng toxins. Kasi heat stable yung enterotoxin. Okay? So ito yung mga toxin that withstand high temperature. Okay? And then the other one is cerulizine. And then the other one is phospholipase. So these are examples. Of virulence factors. Okay, virulence factors of Bacillus cereus. So they can cause gastroenteritis. The other one is this is supposed to be letter A, ophthalmitis, and then opportunistic infection. So if Bacillus anthracis utilizes cleft medium. This one, you'll be using the PIMBA, which stands for polymyxin, pyruvic, egg yolk mannitol, bronchymol blue base. Okay, bronchymol blue. So, if you're using the PIMBA, the colonies appear to be blue, rough, and dry. Okay. Bacillus cereus would infect also hosts that are immunocompromised, meaning you have weak immune system. So, Bacillus cereus could also be a form of opportunistic infection. They can infect your eye, yun nga yung panophthalmitis. They can cause meningitis, can go to your bloodstream and cause septicemia, or worst case scenario, even go to your bone, so that you'll have osteomyelitis. So, they're even found as contaminants in drug paraphernalia. Yan. Yung mga, ano ba yung mga drug paraphernalia? Mga ginagamit yung mga nagsashabu. Diba? Yung pala, mayroong Bacillus cereus. Okay, so, here's how the organisms look like. So, take note of this. So, there is what you call beta hemolysis. Okay, gram-positive rad. So, there are two types of food poisoning that are associated with Bacillus cereus. So, emetic form and diarrheal form. Diarrheal form, and let's discuss first the emetic form. The emetic form is caused by the heat-stable enterotoxin, meaning even if you heat the food, the toxin is still stable, such as rice. Or